Jnanam Paramam Dheyam. Knowledge is supreme.
Welcome to MOOC course on applications of Interatomics using genomics and Proteomics technologies. Today is going to be the lecture in the very exciting areas of next generation sequencing technologies. I am sure you are all aware about the progress what we have made in genomic technologies. In the year 2000, 2001 and especially till 2003, the draft human genome map was published. And along with the draft human genome project when it was getting accomplished, many other model organism sequences were also finished that time. And very first time we got glimpse of the possible genome sequences available for different model organisms. These projects were really long, just imagine it took maybe 12 to 15 years time to accomplish sequencing of one individual and one model organism. After that, looking at its success, looking at the impact of genomic technologies, a lot of innovations happened. This is one of the such an integrated area, and I must say you can appreciate how biologists and technologists and clinicians can benefit from each other is one of this area when genome sequencing information really triggered interest of engineers to come forward and make the new technologies which are much more rapid, much more robust, much more reliable, much more reproducible. And those have resulted into series of next generation sequencing technology from first, second and the third generation technologies. So, these technologies interface have really helped us now to move forward for what was accomplished in 10 years to maybe in a day or two, you can now do the sequencing. So, this is as I said these are the kind of technologies. Sometimes for a revolution to happen you have to wait for decades and you have to wait for centuries, right. But this is the kind of technology which actually happened in front of my eyes and during my career, and I am sure some of you would have also seen and witnessed the kind of progression we have of the old Sanger sequencing based methods and moving toward the very fast rapid next generation sequencing platforms. This is a really interesting area. I think it is a good idea for us together to learn something that what are the current available technology platforms which can use to do the sequencing in a very high throughput manner. And let us also think about how best we could use these for many applications. So, in this slide we thought to provide you couple of technology overview as well as some of the applications from series of application scientists. In this series, today we have Mr. Praveen Nilaway, a field application scientist from Thermo Fisher, who is going to talk to you about the ion torrent informatics solution for NGS data analysis. So, let me welcome Mr. Praveen to talk to you about this novel technology platform of ion torrent, and he is going to then talk to you about the data analysis and applications.
Good morning everyone. So, you must have gone yesterday through the ion torrent technology, right? You have seen about sequencing yesterday, so you have got an idea of how the system works. It is about signals, it is about recording the data in few minutes within seconds, and then you are going forward with sequencing and recognizing which are the bases that are getting generated, right. So, you have seen lots of steps in doing sequencing. Today we will take it somehow to the steps of understanding those sequencing and how it is interpreted into results. Okay. So, I will take it point by point what it is actually. So, we can move forward. So, what we are looking at into NGS technology or NGS sequencing, we are looking into something like where we will have end-to-end workflows or end-to-end analysis to be completed, right. So, you must have heard yesterday about something called as Stargate sequencing, yeah. It is something like your genomics is targeted for particular region, or it may be your genes that you are studying are targeted for a particular region through primers, and you then pick them up and go forward with sequencing. Okay. So, once you start sequencing, That's the first part what we look for, that is the design. So you are targeting particular regions for particular variants. Now you must have heard, why are we going to target those particular regions? So important part is you may be studying certain hereditary diseases, you must be studying something cancerous diseases, right? You must be looking for something genetic disorders into it, right? So those genes which are very much important for you, you like to know which are the regions where exactly those variations are happening, or those are the mutations you want to capture in your study and know at what level those mutations are happening into your cells or into your samples, right? So for that thing only you like to target those regions, right, and take them further for your sequencing. So what happens over here is you have a technology called as AmpliSeq Designer as the first stage, which helps you to design those regions which can be utilized for sequencing in NGS over here. So in this we have in 3 stages. One is the design, second is the analyze, and third is something called as report. Okay, so you try to design your regions of interest which you are looking for your diseases, say hereditary diseases. You take those design for particular genes, you run your samples onto your system called as Torrent suite or else Iron Torrent, and the software that does the analysis for you is called as Torrent suite software. In this software, it will help you to understand what are the variants that you are getting and at what level or which level of coverage are you getting in these data. So you are looking at Torrent suite software where we are taking all the variants in our hands and trying to study them. So with this variants where you are just getting to know that there's some change happening into your gene, you also want to interpret them in a way where you can understand it what exactly is happening to the protein level or what is actually happening at the sample level, right? So you'll do is last part is to report that information through various databases. Have you heard about OMIM? OMIM, right? You have heard about DBSNP, database of SNPs, single nucleotide polymorphisms. Have you heard about anything about cosmic databases which actually hold your cancerous variants? Okay, these are some words that are coming over here. So these are very much important when you try to study certain different diseases or disorders into your analysis. So now what we are doing it over here is you have 3 stages. I'll take each stage individually and explain you as such. So the first part is AmpliSeq Designer. So what we have is a complete workflow of doing the analysis where you like to know which are the regions of interest of yours. Whether I could design my regions in such a way that they could be sequenced on a NGS technology. Okay. For doing that, we have a tool called as AmpliSeq Designer. It helps you to take all your required designed genes in as a list of genes or even the region of interest from your chromosomes. Okay. It takes that and helps you to design primers which could fit into technology of iron torrent sequencing. Okay. So you can design something of your own. Okay. Something like what you do on your websites. Normally you create your own accounts. You can create your own account over here where you can also give the design name to it, and then what you can do is you have certain application types available. So you can see you have the first option is DNA over here. So this is like a gene based design that you can do where you can provide just the Hugo nomenclature genes over here. So you must be knowing about EGFR if you have any idea. You knowing about some BRCA genes, BRCA1, BRCA2. So these are some of the genes which are studied a lot into the entire world. Okay. So they are studied for their different variants. They are studied for their different purposes, where they also want to come down to a place where they can get to know which drugs are acting on regards of cancer, in regards of your hereditary disease. Okay. So at the same time, I have something like the gene design. I also have something called as hotspots. So I was talking about something called as cosmics or DBSNPs. Okay. So these are variants which are already known or people have already studied. Researchers have already known these are actually a deleterious nature variants that are coming out, which are having effects onto your particular patients or group of patients. Okay. So if I know those variants and I would like to check it in Indian population, I like to design a panel in such a way that I'll use those hotspot information over here and I could apply it overall for all the population in India. So I may have 100 to 500 samples. I like to test them overall and get to know whether the same variants are actually falling into your data or not, or into the Indian population or not, right? So this could be very easy for one study that is called as pharmacogenomics, right? Your one gene can bring you results in such a way that it could easily tell you which therapy could be properly utilized for particular patients, right? So over here we have an option of DNA hotspots which take such type of information. It may be a chromosome single location such as an SNP, it could be a deletion of a bigger range. So in that way we can provide the information over here as hotspots. The rest other two things that are available, one is gene expression. So you must have studied about RNA-seq. Have you heard about it anytime? Or something called as whole transcriptome. So this is nothing but a study of genes which are expressing into your samples. Or you can say if you are considering something like you have cancerous patient and a normal patient. So you like to know which are the genes that are expressing into a cancerous patient, which are very much different from what the normal patient is having, right? So such studies come into play when you talk about whole transcriptome sequencing, okay? There's a different technology. At the same time, we can do same gene expression analysis into this place, where we like to target those genes which are actually into your expression studies, okay? And we like to know which are actually highly expressing, which are actually low expressing. This could easily take you to the pathways which are affected due to these regulations, okay? So this is one of the study, and the last one is something called as gene fusion, where two genes can fuse at a particular location, at a point, and there could be a protein change happening into it. So such study or such type of designs could be made ready, available, which could be taken up further for sequencing onto ion torrent technology, okay? So I'll just move forward. So what happens with the design? So I'll just give you a small example of a BRCA gene. BRCA1 gene which has been designed over here. So you can see BRCA1 gene, and then it says what is the DNA types that you want to go for. Now what happens into your data? You can get data primers designed based on different types of samples, okay? Now whatever you do, you can be using, like, you must be studying blood type of samples. Like normally what your lab test does are, you take only the bloods, you take the blood samples, you like to take all the genomic data out of it, target your particular gene and then do the studies further, right? So such type of analysis could be done. We can get a design ready for it, and in that I get to know how many amplicons are getting designed for a particular gene. Now my BRCA1, I think the design looks like you have 36 amplicons that have been designed over here just for your DNA sample, okay? So such type of samples or such type of variations is available over here just to know what can be done. There's one more type of sample that comes up into cancerous studies. Have you heard about FFPE sample anytime? Yeah, these are actually formalin-fixed samples, okay? So these are actually whenever you do cancer test or somebody is detected with cancer, these samples are actually fixed on a paraffin, right? And then sent across to labs for testing. So these can be easily picked up. So you wanted to know what are the variations or mutations that you are getting into it. So such studies could be done again using particular design over here. So what happens, this tools helps you to differentiate between what are the DNA types, what are the type of data that you want to go utilize for doing your studies, what type of targeting that you can do in such type of studies, okay? So once this design is ready, you get to know how many amplicons are there, what type of sequencing I could do. Like the technology is defined based on my sequencing range also. So you have something like for FFP it is 125 to 175 base pair. So I have utilizing a technology or sequencing where I could do 200 base pair sequencing, and there is also a 400 base pair sequencing which I could take further with my blood samples also. So this helps you to easily get to know what things has to be utilized, how could I run the data onto an ion torrent system, and you do further, take it further for the analysis level. So you got to know about the information, how designing is happening, right? How the panels are getting designed, your genes are getting picked up and taking it further. Now once your genes are ready, your panels are ready, you like to take it further and sequence it. And you will put it onto the system. You will first do all the amplification. Yesterday you must have gone through all the steps for whatever the amplification happening, you are taking it, barcoding it, and then taking it onto the chip and running it, and your reports get generated finally, right? So once your reports are generated, the report analysis work is done by this software, Torrent Suite software, okay? It understands whatever data is generated by signals, okay? Whatever voltage changes are happening are recorded onto the systems, and the software understands its signals, clears the signals or filters the signals over there, and then decodes them into particular bases. So you have a chip has millions of wells, and in millions of wells you have millions of signals getting recorded, and the same signals are decoded into your bases and giving you the entire read length, a bigger read length sequence over there, okay? So you get sequences which are around 200 base pair, 400 base pair, or it may be on a higher read length of 500 to 600 base pair also, okay? So once you have done sequencing, what should we do further? We have got the raw data, right? See, this is the raw data comes into various formats. One is FASTQ. You must have not known about it, or you may be having an idea about it. FASTQ files, which is a raw data file containing sequences as well as the quality values for it. And there is something called as BAM file also. So this is just for knowledge. BAM file is one where if you do certain like aligning to the genome, your data is generated and you are aligning to the genome, you get these BAM files which contains all the coordinates for those genomes. So you align it, get the coordinates, which are the chromosome, where is the actual alignment happening, which position, and whether it contains proper alignment or there are any mismatches or deletions into it, insertions or deletions into it, right? So everything is recording through the software, Torrent Suite software. The tool that does this mapping for you is called as TMap. Okay, I have not put much into this. I'll just take to the specific level of analysis that we do. So once the system internally does the alignment with a reference genome, say if you are running a particular BRCA sample and you do a run and you have a reference human genome, you align it. So you like to know what is happening behind it, right? Your genome is getting aligned, your data is getting properly aligned to it. But how? How much is the data getting aligned to it, right? That would be your first question. So if I have generated around 2 million reads for that sample, how many reads are actually aligning to those regions which are interest of yours, right? So for that, we have a tool called as, sorry, I just explained a lots of thing of the Torrent Suite software. Just to take it in a shorter way, you have something like automated sequencing data analysis. You have something like the interface is through a web-based interface, where you easily can go into a website and go through the runs, reports, download the reports in PDF, run different plugins that are available for doing analysis, okay? So as I come back to what I was speaking, you have done data analysis onto a reference genome, aligning your data to that reference, but you need to know something more about it, whether it is actually representing your data or not, actually it is aligning to your regions or not of interest, right? So that's my main important point. So over there are helpful, whatever plugins are there, those come into play over here, okay? So we have certain plugins which helps you to understand if the region of interest that you have designed, I could provide my information over there. Say I have BRCA1 region, I have designed it, I wanted to know how many genes or how many reads are actually aligning to those regions, okay? So how can I do that? So I have a plugin called as coverage analysis, okay? This coverage analysis helps you to know if any region is getting aligned with number of reads, it lets you know how many reads are actually mapping to it. So what happens? So it says if I have a region, say this is an example for a cancer hotspot panel, which has around, in the target region, has around 22K bases, 22,000 bases into it. So I just wanted to know how many percentage of bases are, like how many reads of my, or how many bases of my reads are actually on target, aligning to my BRCA region or like CHP panel region. So it is 90% of my bases are actually aligning to my region of interest in cancer hotspot panel. Okay. With that, it lets me know which is the base depth coverage across it. This is like if I'm aligning my reads or my sequences to a particular region of interest, how many reads are overlapping in those particular region. So what is the mean depth across those particular region, right? So I need to know if a read has been covered by how many reads are covering a particular base over there. So in that, you have around 9,495 mean depth. Okay, it's an average depth covering a particular base over there. So you may be having a range of around 8,000 to 10,000, okay, into your CHP panel that have you designed, or it may be your custom panel that you have designed, and then you get to know this particular depth. Now this gives me an idea whether my panel is properly having all the reads or not, right? If I am having a depth of 1,000, 2,000, it's still good. So now over here as an example, it's very high. And that I'll also like to know whether my region of interest, say I have 22 KB of my region of interest, in that whether if I look for 1x coverage, how many bases are covered by 1x coverage, okay? At least one read covering each base. That's what I could say as 1x coverage. So in that you have around 100% bases which are covered at 1x. At the same time, I like to know how many bases are covered at 20x, that is 20 times a particular base is covered properly or not, right? So 20 times a read is covering a particular base. So at the same time it's 100%. Till the 500x you can see it's 100%. So this shows me whether my design is fine or not, whether my each base is getting covered properly or not, whether this data could be taken further for my variant analysis or not, where my variant of interest would be properly picked up or not. So this gives me an overall idea. At the same time, I have something like end-to-end reads. So if I'm designing something like your gene, okay, I'm taking my gene, designing a particular primers for your exonic regions, one end to the other end. So whether it is covering end-to-end or not, so how many reads are actually covering end-to-end sequencing into it, right? That gives you a confidence for your primers also. So your primers which have been designed, so it shows me 95% of my total reads are actually having end-to-end alignment to my region, gene of interest. Okay, so that gives me more confidence whether my data is coming good or not. Okay. So any questions till now? Yeah.
Does this 1x coverage essentially means that single base resolution?
It's not single base resolution. It is like when you are taking a reference genome, you are aligning a sequence. I'm just trying to know whether a single base is covered by one read or not. Okay, so I'm aligning. Alignment is nothing but you have a reference, you have a read getting aligned to it. Okay, there may be 1,000 reads that are getting aligned. So I'm just trying to get to know whether that particular base in genome is getting covered by how many reads. So if I am having something like one read passing to it, so it is covering that particular base. Okay. So that is the first stage actually. Once that is done, it is taken further and aligned to the reference genome. So in alignment, what happens, we like to see how many reads are aligning to a particular region. And the same thing I like to calculate over here. We need some statistics, right? We can't go and visualize the data every time. There is a way to visualize it. The tool is called as IGV. So this is called as IGV, Integrated Genomic Viewer. It helps you to visualize your data, how much data has been aligned to that region, how many data is for other regions. So you can go scroll through genes to genes and get to know that. Okay, this is just an example that I've put forward. One of the gene which has been aligned by the reads, it has the forward reads as well as the reverse reads into it. Okay, so in this way we like to know whether the regions are getting covered or not, whether there is a proper coverage coming at my gene of interest or not. Okay, so after this, what I will be going to do forward. So I have another plugin called as Torrent Variant Caller. Okay. This Torrent Variant Caller is optimized to calling all my variants. So my variants could be SNP, indels, right? So these variants could be called using this software, Torrent Variant Caller. It has features into it, such as I could give the chip types. Since the system over here right now is Ion S5, we still have two more systems available. One is Proton and one is PGM. Okay. So after that, you have almost similar kind of workflows. So you can go for CHP panels. As I was saying, you have different panels available for cancers. You have panels available for hereditary diseases. So with that, what I get to know is there is something called as design files. Okay. So whatever design that you create on AmpliSeq, those files could be downloaded in a particular format called as bed format. And these files could be uploaded for doing the analysis in Variant Caller. Okay. Once my design is uploaded, variants would be only called into those regions which we have designed. It will not go and look for some other regions. Okay. Once those designed region is given, it also has something called as hotspot. I had spoken about earlier about designing hotspot regions also, where I could like to know whether any of my known variant is present or not. So I could give a file called as hotspot.bed file in a format such a way that it will recognize that particular position into my data, into my variants, and it will then represent it that yes, this is the hotspot that I was looking at. It will provide you the final results in an Excel sheet. Okay. So what you do is further, I give forward my designs, my hotspot regions of interest, and I could give certain parameters to call the variants. So variants could be called based on my somatic nature or else my germline nature, 50% frequency or else 5% frequency. And then the variants could be called at that stage. Okay. And once I run this plugin, I could submit the data, and once I run this data, I'll get all the variants that are generated into it. Okay. So what happens? The variants could be downloaded for all the SNPs indels into an Excel sheet entirely. So you can take that and study it further. So now what is happening over here is, so I'll take a step-by-step mode. I first did designing on AmpliSeq, right? Then I did the Torrent suite software where it decodes all the bases and provides you the sequences for it. It provides you alignment with the reference genome. It does the coverage analysis for you, which looks for the regions which have been designed, your interest, gene of interest, and then take it further and do variant calling. So once you have done with the variant calling, you just have variants with you, right? You have just the SNPs giving you the change from A to T, C to T, or just the deletions, A is deleted or T is deleted. But you still need to know something more about it, where exactly this is happening, which gene it is happening, whether it is actually having deleterious effects or not, whether it is having any effects with the patients or not, right? So you need to know something more about that. So for doing that, there's one more tool called as Ion Reporter software. So it has lots of information inbuilt into it. So this helps you to correlate your variant with the information that is already stored into databases. Okay.
So today you learned and at least got a glimpse and some understanding the basics of NGS platform and data analysis. You are also introduced to the Ion AmpliSeq Designer, the Torrent suite software and the Ion Reporter software. While some of these are very specific to a given technology, which is not the mandate of the course to teach you how specifically these softwares work, but rather by showing these kind of available platforms, intention is to give you an overview and a good understanding that how these technologies and these softwares could be used for your applications. So in today's lecture, you also got understanding about how to visualize the data and interpret the NGS data. Usually when we are able to obtain this kind of big data set, I think it's really important to look at data in really systems wide manner, right, and the big data being generated, and your intention is also to integrate the data and compare data from other systems as well. So, in this light, having dedicated server and high computing systems can definitely help to do the analysis in a much more rapid manner, and that could also provide us to do lot more things which one could try to do now, comparison, aligning with the reference genome, and many other type of multi-omics analysis can also be performed. So, I must say that one thing which is limitation is our computing power, the way we can process the big data simultaneously for large number of samples, as well as different type of information obtained at the gene level and the protein level or mRNA level, and trying to correlate all that information together. We need really the high computing power and lot of space to do these kind of analysis. So, next step is to do variant analysis, which can be also done using a cloud based tool, and I am sure in the as we go along in the next lectures you will specifically study how to use this data for more application orientation, especially in the context of cancer. So, we will continue more on this slide of NGS and its revolution, and we will talk to you again in the next lecture. Thank you.